How did you come up with the idea of migrastatics?
There is an analogy with cytostatics. So cytostatics are drugs interfering with cell proliferation, while migrastatics are drugs which prevents invasion and metastasis of cancer cells. We have analyzed the behavior of Fibrosarcoma cells, which use both Amoeboid and mesenchymal modes of invasiveness, and we found that their behavior is much more plastic than we expected before. The cells which are using Amoeboid mode of invasiveness could use the mesenchymal cells tricks and vice versa. This could help us to refine our subsequent research, which is concentrated on development of drugs inhibiting both modes of invasiveness.
Can you share with us the most interesting research findings so far?
We have contributed significantly to the understanding of both mesenchymal and Amoeboid modes of cancer cell invasiveness, and in collaboration with medicinal chemists, we started to produce lead compounds for the development of migrastatics.
What cell parameters or features are of interest to you?
We are mostly interested in translocation of matter in both mesenchymal and Amoeboid migrating cells, but also QPI enables us to observe cells in environment with scattered light.
How is Q-phase helpful in your research?
Q-phase help us to understand in detail both protease-dependent and protease-independent invasiveness of cancer cells.
Do you combine QPI imaging with any other imaging techniques?
We combine it mostly with confocal microscopy, which we use kind of as a medium contact screening for interesting behavioral situations, which we then analyze in detail with QPI.
Can you describe this image you took using the Q-phase?
On this image you can see Fibrosarcoma cancer cell, and the cell is trying to move forward, but the nuclei of the cell, which I am showing in the middle of the cell, is squishing. To go through the collagen network, the cell is starting to make blebs, which are kind of rounded protrusions around nuclei, which is the way the cell is trying to remove the collagen fibrous obstacles around the nuclei, and which should enable her to move forward. This is the same type of cells, Fibrosarcoma cells, which are basically cancer of soft tissue. And here you can see another very interesting aspect. The cell is sticking to the collagen fibers, which are depicted in colors. And as you can see, during the time, it is moving the fibers and joining them together to make a stronger fibers out of the individual fibers, which is helping her to move forward. On this graph, you can see actually the cell divided into leading, retracting and side part, and you can see on y-axis mean mass density, and by that you can see that this graph documents that front parts of migrating cells are heavier, which facilitates their movement, and we observe that for both Amoeboid and mesenchymal cancer cells.
What is this video showing that you took using Q-phase?
Cell is moving through the 3D collagen matrix, and you can see the beautiful blebbing around the squished nuclei of the Fibrosarcoma cell during the movement.
Would you recommend Q-phase to other users?
I would definitely recommend Q-phase to researchers in the field of cancer cell migration and invasiveness, particularly to those researchers who are interested in analyzing cancer cell migration and proliferation advance, and also which are interested in translocation of matter within migrating cells.